Hey there, everybody. Just getting started today on Wednesday, what is it, February 4th, and now thanks to all of our crazy snow days, we are completely off schedule. But that's okay. We can roll with the punches. So what I'm going to be doing today is I'm going to be going through what's happening next week in the lab. So because we didn't have lab on Monday and Tuesday, we're not going to have it Wednesday and Thursday because we don't want everybody to get all off schedule, and we're using produce that can go bad and get yucky by next week. So we'll be doing 2 labs next week. So it's extremely important that you come to lab next week because we're going to be having 2 labs, and then the week after that you'll be having 2 quizzes. So make sure that you're coming next week because that's going to be extremely important. If you miss 2 labs, that's double the points. So in getting started today, I'm going to go to our web page that has all of our information on it. You can do the same thing. You can go take a look at our schedule, which obviously I'm going to have to adjust now because we did not have the Lab 4 here. So we'll be having Lab 4 and Lab 5 February 9th through 12th. So the 2 labs that the lab we would be doing this week and the lab we're doing next week. The lab that we would have done this week is Lab 4 on enzymes and catalase, which I'm going to go through with you here. And Lab 5 would be DNA extraction from strawberries. So here's my general plan. I'm going to do the lectures for Lab 4 and Lab 5. That way you don't have to spend time on those in class. So when you come in next week, you can automatically get started in doing both of those labs together. I'm also going to talk to you a little bit about the group projects, and this will be something that you'll need to do next week, so you will need to bring a laptop with you. So basically I'm going to write that down because I have all these fantastic ideas, and then I will totally forget what I'm doing if I don't write them down. So I got my handy dandy little calendar right here. I'm going to remind students about laptops next week, and I'm going to see, I already don't remember what I said I was going to do. I'm going to adjust the syllabus on big picture biology, and what was the other thing that I just said I was going to do? I don't remember, but thankfully I got it all on video, so I'll be able to go back and take a look at that. So let me go ahead and get started. What's it about the group project? Okay. I guess you guys can see how easy it is to get distracted. So we're going to be looking at Lab 4, which is enzymes. Excuse me. I've got the hiccups now. And as we pull that up, you can see the PowerPoint presentation, which is right here. So let me go ahead and get that started. I'm going to go full screen, and let's go ahead and get started on that. So in Lab 4, talking a little bit about enzymes and catalase, we are going to be looking a little bit at hydrogen peroxide. Hydrogen peroxide, the chemical formula for that is H2O2. So you'll see that it's, even though the chemicals are similar to water, it is definitely not water. We're going to recognize different types of enzymes and their characteristics, which will be interesting as we talk about spring approaching in March and April, about why enzymes are important to things happening in the spring. We'll be looking at catalase in animal cells versus plant cells, and we're going to explain why temperature and pH would have different effects on enzyme activity. So let me get a sip of my coffee. What are enzymes? Enzymes are something called biological catalysts. Whenever you hear that word catalyst, I think of something that energizes something else. So if you are a catalyst for change, like we heard in the presidential elections, a catalyst for change, you're something that is helping change to happen. So enzymes are biological catalysts. They are substances that increase the speed of chemical reactions. So naturally, inside of your body, you have all kinds of chemical reactions going on. As you just saw me drinking coffee, you can imagine the chemical reactions that are happening to my body as I'm getting the water from it, as I'm taking the caffeine from it, as I'm going through metabolism. And so when an enzyme is present, it takes less energy to start a chemical reaction. So you've got all these chemical reactions that are happening within your body, and normally they would happen pretty slow. That's why our body produces enzymes in order to increase or decrease the activation energy, make it take less energy for those chemical reactions to get started. And so enzymes help us maintain homeostasis. Inside of your body, you've got this natural balance, and that is homeostasis, where your blood sugar is right, your temperature is right, your pH is right, and if at any point in your life your blood sugar is not right, then that's a problem. If it's too high, that causes certain problems. If it's too low, you could pass out. You have a certain temperature in your body. If your temperature gets too low, you start to shake and get the shivers and goosebumps, and you could go into hypothermia and die. If it goes the opposite direction, if your temperature is too high, then you may have a fever, and if your fever gets too high for too long, then it can cause the proteins in your body to denature or break apart, and if that happens, you're toast as well. So our bodies are really amazing in that they're struggling to keep us at this homeostasis. There's this battle going on within your body to make you at this even kilter. So it's pretty amazing that our bodies actually do that. We are complicated beasts. And so enzymes also facilitate metabolism. So metabolism has to do with you taking in vitamins and minerals and food macromolecules like lipids and starches and sugars and proteins, and you using those things for energy. And if you weren't eating, if you were to starve yourself, eventually your cells wouldn't have any energy to go about doing their processes, and cell death eventually equals you death. How delightful for this morning. So what are enzymes? I think that enzymes are something that's really difficult for students to understand because it's one of those words we don't really use that often. Unless you are a diet guru, you may not be thinking about the enzymes that are constantly at work within your body. So what are enzymes? Enzymes are proteins. If you look at the picture there, you can see they are 3D proteins. They are this twisted package of amino acids. So lots of A's and C's and T's and G's put together to make these large proteins. And these proteins are going to, 1, be the building blocks of you, and 2, tell the you how to function. So it builds up your body, and then things like hormones and steroids and enzymes are going to make you do the processes you need to do in order to be alive. So each one of the enzymes that are inside of your body are going to work with a very specific substrate. That means the molecule that's acted on by an enzyme. So the enzyme is this protein that acts on a molecule in order to speed up this chemical reaction. And so we'll be looking at this a little bit more as we go along. I hope my hands don't look all crazy. I'm not sure exactly what it looks like, but I guess I'll have to go back and watch this video. So here what we see is we have an active site. So our enzymes are basically like a lock and key. They've got this very specific fold to them, and they fit into a substrate to form what's called the enzyme substrate complex. So an enzyme plus a substrate are two little puzzle pieces that fit together perfectly in order to form this enzyme substrate complex. So you can see it's folded in a certain way that it matches up exactly, fitting together like a puzzle. I also think of it sometimes, because I'm diabetic, I think about insulin and the way insulin acts on the sugar molecules. It's like a lock and key. The insulin unlocks the sugar molecules, making them available for energy sources within my body. If I don't take insulin, I'm diabetic, I'm type 1 diabetic, if I don't take my shots every day, then basically my body starves because it can't get any of the energy from the sugar. So you can see it's a very complicated process, and if anything goes wrong, then you can end up in trouble. So enzymes always match up to a specific substrate. If you look at the picture right here, you can see that the first enzyme that we have is a lactose molecule, and lactase is the name of the enzyme that fits together with lactose and makes that milk sugar available. In the one on the bottom, you can see a starch molecule, and it is using the enzyme amylase to unlock that energy that's within the starch. And so if something goes wrong, like if you can't digest lactose from milk, like if you're lactose intolerant, sometimes you can take a enzyme called lactate. You've probably seen it in the grocery store, or if you've ever heard of lactate milk, it's like milk that doesn't have milk in it, and it makes it so that you can drink the milk without getting sick. People that are lactose intolerant get really sick eating milk products. The starch and the amylase is what's found inside of your mouth, inside of your saliva. So if you've ever done the saltine experiment, where you basically put a saltine in your mouth and let your saliva go about starting to digest it, because digestion starts in your mouth with your saliva. So you put the saltine in your mouth, and as your saliva starts to mix around with it, you start to taste a really sweet, kind of sticky feeling happening in your mouth. That's because the sugars are being released from the starch as your amylase in your saliva starts to break them down. And so your body goes about all these amazing things to get the energy that it needs from the food that you're eating. So each enzyme has this definite 3D shape. You can see that the lactose and lactase look different than the starch and the amylase. So different 3D structures for all these different enzymes in our body. And each enzyme is going to act on only one specific substrate. So it means lock and key. That key fits with that lock and only with that lock. It's only going to break down specific substances. So what do we need enzymes for anyways? We talked about breaking down starches in your mouth. We talk about breaking down milk sugars. But enzymes do things that you probably have no idea about. In order for the leaves in the fall to turn the beautiful colors, here we are in Ohio, we've got yellows and oranges and reds and purples. Enzymes work to turn the colors, the leaves, those colors in the fall, and then they fall off the tree and become part of the dirt. The enzymes are also helping to ripen foods. So if you've ever seen your bananas go from beautiful and yellow to mushy and brown, that's enzymes at work. Or if you've ever had an apple that you pressed on it, it's got a big mushy spot, that's because enzymes are going to work breaking it down. We've got the digestion of food happening within our bodies all the time. Enzymes are going to help to produce DNA and produce hormones. And so without life, or without enzymes, life would not be able to happen. Without enzymes, the chemical reactions inside of our bodies would happen so slowly, we would not be able to survive. And without enzymes, our food wouldn't be available for us to eat. The plants that are out there in the environment wouldn't do the things that they need to do. And so without enzymes, life as we know it would disappear. That sounds really gloom and doom, doesn't it? So what are some things that affect enzymes in a reaction? So thinking about as the temperatures get cooler in the fall, the leaves are going to begin to change colors, and so temperature is one of those things that affects enzymes. Every enzyme has an optimal temperature. The ones that are inside of your body are going to happen best at 98.6 degrees Fahrenheit, 37 degrees Celsius. So the temperature in your body is going to affect enzymes. The other thing that affects enzymes is pH. So how acidic or how alkaline it is inside of your body or out in the environment where these enzymes are. So pH is going to affect it. So that should make sense. Temperature around you, amount of acidity. So here, looking at temperature, enzymes always have one specific temperature where they're optimal. So in the human body, 37.5 degrees Celsius. So they work slowly if you're cold, and they work too fast if you're hot, like if you have a fever. So what that's called when you have a fever and your enzymes begin to break down, that's called denaturing. So make sure that you remember that word, denaturing. Then we have everything having an optimal pH. Inside of your body, there's a little bit of variability because if you have enzymes in your stomach, they're going to work at a different pH than if you have enzymes in your blood. So the enzymes in your stomach work at a very high acidity. Your stomach acids are have a very low pH, and so the enzymes that work in your stomach are going to be different than the enzymes that are found in different places in your body. So all enzymes have a specific pH where they work the best. And we basically already said that. Low temperature reactions happen slowly. High temperature reactions happen so quickly that eventually those proteins can denature. Remember, your pH scale goes from 0 to 14. The pH of pure water is 7, and the pH of your body, I believe, is about 7.3. And so we are a little bit basic. And so changes in pH can also change the structure of proteins and how they function. And so organisms can only withstand a very small fluctuation in pH. If something changes your body's pH, then things start to go out of whack. So here we have a couple of enzymes and their substrates. We have starch molecules, which are complex sugars, being broken down by amylase, which is found in your saliva. Maltose is a specific kind of sugar that's broken down by maltase. Sucrose is the sugar that's found in a lot of your foods. I was just reading my yogurt this morning, and it has added sucrose, so they put added sugar in it, which is broken down by sucrase. We've got lipids broken down by lipase, proteins broken down by pepsin, and those are kind of interesting because if you think about things like meat tenderizer, if you pour the powdery meat tenderizer on your meat, it makes them softer because it's basically beginning the process of digestion for you. And that meat tenderizer, I believe, is made out of a enzyme called bromase or bromase. It comes from pineapples, so who would have thought pineapples help you to begin digestion of your red meats. And then the one that we are going to be working with in lab next week is catalase, and it is going to work on hydrogen peroxide. So yes, we're talking about the hydrogen peroxide that you find in your medicine cabinet, and that's what we'll be working with next week. So catalase, what it's actually going to do is break down hydrogen peroxide inside of your body into 2H2O and O2 gas. So maybe your experience with hydrogen peroxide is somewhat like mine. So if I were to get a cut on my skin, I might take and put hydrogen peroxide on it, and I'll start to see it bubbling, and that will clean any bacteria out of the wound before I then go about covering it up and put a bandage on it. So that way I'm not going to get infections in the cut that I have. And so what it's basically doing is the hydrogen peroxide is breaking apart into its contents, into water and oxygen gas, so hence why you see the bubbles. So the products of the above reaction are oxygen gas and water, which are both not poisonous. What's kind of funny is that water and oxygen gas are things that we need to be alive, but hydrogen peroxide, which is just water and oxygen gas put together in a little different way, then that becomes poisonous. If you were to drink hydrogen peroxide, you would be in deep trouble. But in living cells, we use water and oxygen, the oxygen for cellular respiration, and the water is excreted. So take a look at that chemical shape down there. The two oxygens bound together, and then the hydrogens bound to those oxygen. So H2O2, even though it's very chemically similar to H2O, hydrogen peroxide is toxic, and water is not. So just by changing the chemical formula, changing those the way that those molecules are bound together, we are changing a lot about those chemicals. So an extra atom of oxygen makes hydrogen peroxide a very different chemical than water, and it can be dangerous if it's concentrated. So, like for example, when I was getting out the hydrogen peroxide to use for this lab, on the outside of the bottle of hydrogen peroxide, some of it had concentrated, and when I put my hand on the bottle, I started to feel my hand tingling, and when I looked down, my skin was kind of a crusty white color because the very concentrated hydrogen peroxide was killing the cells on my hand. And so even just a little more concentrated than 3%, that is very dangerous. And so the hydrogen peroxide that you get, like I got it at Giant Eagle, it is extremely diluted. If you use stronger hydrogen peroxide, ladies, if you've ever gone to get your hair bleached, it is going to bleach out the color of your hair. Another thing that it's used for, this is kind of maybe gross to you, is that if you ever go to a taxidermist, taxidermy where they take dead animals and they mount them to look like the real animals again, if they're going to bleach out the skulls so that they'll be white so they can mount them, they do that by boiling them in hydrogen peroxide, and so that is really a disgusting process, and it's also a dangerous process. So today in lab, or well, next week, usually it would be today, sorry, I get so used to saying things. The scientific terminology that we're going to use in this lab are things that we're going to use throughout lab this entire semester. And you should know what these three things mean. You should know, first of all, what the control is. So the control is something that is constant between all of the experimental groups. And then independent variables are the variables that we're changing between experimental groups. And the dependent variables are the things that we measure. So as we are doing this experiment this week, next week, as we're doing this experiment next week, we will be keeping one variable constant so that we know that that is not changing, and then we'll be changing little things like changing temperature, changing the pH, changing the type of tissue. And as we change those things, we're going to measure to see what is happening in this experiment. So in this case, we're measuring the amount of bubbling that we see in this experiment. And so after lab, you're going to be asked to answer some questions like, what is causing the bubbling to occur? Or why did some substances bubble more than others? And we're going to be using a scientific metric of comparison. One thing that's important for scientists to be able to do is to quantify the amount of things that we are measuring in the experiment, so that if scientists in the future want to do this experiment themselves, that they can say, you know, oh, I also got thousands of bubbles, or I also got too many to count. It's important to be able to have that metric of comparison. Otherwise, what one scientist says is a lot of bubbles may be completely different than what another scientist says is a lot of bubbles. So we try to make things as accurate as possible so that we know we're talking the same language. We're doing the same experiment. And so here we are now at the at the end here. I escape out of that, close out of that one, and let me look back and see how we are doing for time. Okay, 24 minutes sounds about appropriate. So let's go on, and we will do the PowerPoint from Lab 5. I think that when I do this video, I'm going to do a little bit of video editing when I'm done. I'll probably split it up into a couple of short videos so that if you want to watch them, like 4 little short 10-minute videos, or you can watch them all together. It's totally up to you. But let's go through and talk about DNA extraction from cells. So you can start to see some of the major concepts that we're talking about here in biology. Talk a lot about homeostasis, about things maintaining equilibrium or being balanced. Once things get out of balance in nature, that's when stuff starts to break apart. We talk a lot about controls, making sure that we are keeping things constant in experiments, so that, because if you change a whole bunch of variables, you never know which variable it was dependent upon. So the control is the thing we always keep the same. So we have then our independent variable, the thing that we change every time, and the dependent variable, the thing that we measure. So we'll see that in this experiment too, and I will point that out for you as we go along. Get some more of my coffee. Maybe I start talking faster as I go along with this coffee. Okay, skip the text message for right now. Alrighty, let's make that full screen. So today, after completing this activity, you're going to be able to see the physical properties of DNA. We are going to be filtering an extract of strawberry and chicken liver DNA. Yes, both of the labs that we're doing, both Lab 4 and Lab 5, have to do with liver. 2 different types of liver. The enzyme one is using beef liver, and the DNA one is using chicken liver. And this is the last time you'll have to deal with liver. Liver is disgusting, so that's good. So what we're going to be doing here is preparing a filtered extract of strawberry and chicken liver DNA, demonstrating that DNA is a 3-dimensional structure. You can find all kinds of really cool YouTube videos about the about DNA and the properties of it, and I'm sure your lecture professor will be speaking with you extensively about that. So we can talk about how it can be extracted from nearly any cell. We will recognize DNA as the source of variation, so everything is different because it has different DNA, but some things are more the same because they have similar DNA than things that are long-distant ancestors because they have different A's and T's and G's and C's, and DNA is the common link between all living things. So our buddies James Watson and Francis Crick were the first two, I believe, physicists, scientists to discover DNA in 1953. And DNA stands for deoxyribonucleic acid. And you can see before there were ever microscopes that were strong enough to actually look at DNA, they figured out the chemical composition of DNA and how those molecules would have fit together to make this 3D structure that is called the double helix. Basically, it's a ladder with the rungs in it, and it's attached together, and because of the way that the A's and T's and G's and C's fit together, that makes the ladder shift, so it's this double helix. Hopefully you enjoy my hand motions. Maybe I have to edit that out. Parts of the DNA molecule, the sugar and the phosphorus are what makes up the sides of the ladder. They are the backbone molecules. They fit together in little units. And then the nitrogenous bases are the A's and T's and G's and C's, and they make up the steps of the ladder. And they fit together, and whatever sequence they're in is what determines what your genes are. So here we can see that double helix, the twisting ladder, and deoxyribose, that's the name of the sugar in DNA. So deoxyribose is the sugar, And the nitrogenous bases are the A's and T's and G's and C's, and the way that they fit together. So I think if you look up at the top picture of the DNA, that one's the best, because in the other two you can't really see that A's and T's and G's and C's are separate. But in the top one you can see pink always goes with purple, yellow always goes with green, so A always goes with T, and G always goes with C. Those are the fundamentals of DNA. You always know that. A goes with T, G goes with C. And the helpful little hint that I use to always remember that is Austin, Texas is a great city. A goes with T, Austin, Texas, and G goes with C, is a great city. Austin, Texas is a great city. And since I from Texas, I love it. So what I just said, A goes with T, G goes with C. So thinking about the way that your DNA comes together, information is stored in those A's and T's and G's and C's, which make up your genes. And your genes are each going to code for the recipe for certain types of proteins. And as your body is going about making those proteins, your proteins either act by themselves or they are going to act together and perform cellular functions. So they're going to build up the structures of your body, or they're going to make them function the way that they're supposed to. So anatomy, which is the way your body is built, and physiology, which is the way that your body acts, the things that your body does. So it's often helpful to think of this from either biggest to smallest or smallest to biggest. So let's start out with biggest to smallest. So first we look at the human body. And the human body has all kinds of organ systems within it. It's got your nervous system, it's got your endocrine system, it's got your gastric system, it's got your muscular system, it's got all kinds of systems, and that's going to make your body able to function. Each one of those organ systems are made up of specific organs. So your stomach, or your intestines, or your pancreas. So let's look at the stomach. Your stomach is made up of specific tissues. So the tissues in your stomach are different than the tissues in your liver, are different than the tissues in your brain, and those tissues are each made up of specific cells. And so cells are different all around your body. Inside of the cell there's the nucleus, and the nucleus has the chromosomes in it, and chromosomes are made up of genes, and they are wound around something called histones, and what's wound around those histones is DNA, and DNA, when you break it down, is made up of molecules. So we go from body to organ system, to organ, to tissue, to cell, to chromosomes, to genes, to DNA, to molecules. So molecules are the smallest, make up the DNA. DNA is a little bit bigger, makes up the proteins. Proteins are makes up the chromosomes. Chromosomes build proteins, which make up your cells. Cells build up the different types of tissues. Tissues build up the organs. Organs make up organ systems, and organ systems make up you. So it's really amazing to go from this huge human working body to all of these little complex parts. It's fascinating the way that that all works together. So in order to understand who you are, we often look at your chromosomes. So if you were to look at one set of chromosomes inside of any of your cells, you have 23 pairs of chromosomes. You've got 22 autosomes, which are the chromosomes that make up your body. So you can see the first, we put them in order from biggest to smallest. So chromosome pair 1, one of those chromosomes came from your mother, one came from your father, and they are the biggest chromosomes. Chromosome set number 2, one came from your mother, one came from your father. They're a little smaller than chromosome set 1. Then 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So from biggest set of chromosomes down to the smallest set of chromosomes. And then we get to set 23, which are your sex chromosomes. So we've got your XX if you're a female and XY if you're a male. So one of those X's came from your mother because she's got two X's, and that Y came from your father if you're a boy. If you are a girl, the other X came from your father. So very interesting to see the roadmap of your chromosomes. And so the way that each organism has these different assemblages of DNA sequences, this is what makes for variation within all organisms. Because by having a little bit of variation, build proteins just a little bit differently, build these, which build up these enzymes, build up these hormones, and these are going to make each person act a little bit differently, or their body act a little bit different. And these things control growth and development, they control our physical traits. So proteins, amazing things that build up our physical traits, our metabolic traits, and control our growth and development. So here we've got the DNA, which is found in the nucleus of almost all of the cells in your body. So you can see this is an animal cell, and inside of the animal cell, at the heart of the cell, we've got our nucleus, and inside of the nucleus we have our DNA. And the DNA is wound up really tightly inside of the nucleus until the cell is ready to divide, and then it starts to separate out into those little characteristic X's that are the chromosomes. Notice this is a regular animal cell, any part of your body cell, except it is not a red blood cells, because red blood cells lack a nucleus and thus they lack DNA. So looking at the amounts of DNA that are in different types of organisms, you may not think, well, you may think, oh, the smaller the organism, the less DNA it has. Since humans are really big organisms, we must have lots of DNA. That isn't actually true. A lot of bacteria, very simple, have a small amount of base pairs of DNA, but you can see algae have more than fungi, worms have more than algae, crustaceans have more than worms, insects have a large variety of the number of amounts of DNA that they have. Birds have a small variety. You can see mammals down there have a very small variety of the amounts of DNA, but a bony fish has a huge, wide variety. And then if we get down to the bottom, flowering plants, they can have a huge amount of differences in the amount of DNA that's inside of those organisms. So if we were to look at this figure, the DNA that's coming from the chicken liver, we've got our birds with 10 to the 9th power 10 to the 9th power amount of base pairs of DNA. And we've got our flowering plants, which strawberries are a flowering plant. They are the fruit of the flowering plant. They could have anywhere from 10 to the 8th power to 10 to the 11th power base pairs of DNA. Like millions and billions and trillions and more of DNA. So strawberries are going to have lots and lots and lots of DNA, and chicken liver is going to have less DNA. So we might think chicken liver would have more because they're more advanced. That's not actually true. The amount of DNA found within the cells is based on lots of other things besides how complex the organism is. So you might be asking yourself, why should I care about DNA? The reason DNA is important is because every living thing has DNA inside of it. That is the thing that connects us to every other living thing on this planet. The same 4 base pairs, the same A's and T's and G's and C's are going to make up every living thing that's out there. So The difference between you and any other living thing is just combinations of A's and T's and G's and C's. So one of the cool things that DNA can help us do is investigate our ancestors. So if you took some blood from you and some blood from your cousin, you would share a lot of DNA in common. If you are the mother and you have a child, there's going to be lots of DNA in common between you and your direct offspring. So DNA can help us to understand genetic predispositions, like are we going to get heart disease? Are we going to get diabetes? Are we going to get cancer? And it's also going to tell us some interesting things about the way that we act, the way that we live, because our unique DNA makes us who we are and makes us do the things that we are going to do. So in lab today, we are going to be looking at DNA molecules, and we're going to first look at it in strawberries and then look at it in chicken liver. And because DNA is so small, and because we don't have an electron microscope here in this classroom, I actually don't even know if we have one on this campus, but looking at DNA is very, very difficult because it is, you need an electron microscope to see it. It's microscopic. So if you put 300,000 of those long strands of DNA together, it would be about as thick as a human hair. And so what we're going to do today is we are going to get the DNA out of the different types of cells and collect large amounts of it, enough that it is visible. And so we will be purifying the DNA. So here you can see a large amount of the stringy DNA fibers from this strawberry that we're going to be collecting. So in order to purify DNA, just like if we were going to clone an organism, we have to first get pure DNA. DNA is insoluble, so it means it does not dissolve in alcohol. So when it's mixed with alcohol, it precipitates out of this solution, so it comes up out of the solution. Since it doesn't mix with the alcohol, it's going to form a thin layer between the alcohol and the broken apart strawberry cells. And so when we precipitate it out of the solution, we're going to remove small molecules like the salts and the sugars and amino acids that are found left behind in the gunk that is not the DNA. So looking at what we know about most animals are diploid. Diploid, di, meaning two copies, so they have two copies of the chromosomes. Just like we saw in the karyotype, we have two copies of 1, two copies of 2, two copies of 3, two copies of 4. But strawberries are octoploid, means they have 8 sets of chromosomes. So you'd have to picture 8 of number 1, 8 of number 2, 8 of number 3. And what we're going to be working with today is liver. And liver is unique because it is polyploid, meaning it can have lots of different copies of the chromosome 1, chromosome 2, chromosome 3. And so that is the end of that PowerPoint there. So let's see where we are on our recording. I am at 43 minutes. So a couple things that I want to tell you before we go. What we were going to do this week, and which we will do next week, so you have to have a laptop with you, is we are going to be looking at the group presentations. And we are going to start putting together our Google Doc for our group presentations. So I created a fun little video, as you can tell I like to make videos, about how to create and share a Google Doc. So next week when we're doing that, it will be very helpful to you if you have a laptop or if you have a tablet with you. That will be very helpful in creating and sharing your Google Doc. I actually have an app on my phone, and I will show it to you. Let's see if I can do this so that you can see it. Here's my phone, and here's my Google Docs. and look at all those Google Docs that I have. So I can open up my Google Doc for the presentations. It's hard to do this in reverse. And I can look through and I can see all of my Google Docs. And so if you download Google Docs on your phone before you come in next week, that would be really helpful for you. And this is a really cool app anyways, and I use it every day because I keep my, it says, enjoying docs? Yes, write it down. No, send feedback, dismiss. I am not rating it right now. I love it how they pop in the middle of me actually trying to do something. I keep my to-do list on my Google Docs, and that way I can essentially keep a running diary of everything that I'm doing, and it's really awesome for me. Keeping a running journal of what I'm trying to remember to do is super helpful for me, and that way I can always look back at things that I'm like, oh hey, this student told me this one day, or this student athlete is not going to be here this next day. I keep a running list of all that stuff, and I can always find it because I can always search for it on my phone. So if you want to do that, I think that's a fabulous idea that will help you to stay organized. But we're going to do a Google Doc that is shared for your group, and you're going to share it with your TA, and then your TAs are all going to share them with me. So we're going to start working on putting together our presentations, and I'm going to go ahead and shut this off here, and I or your TA will see you next week. I hope you have a wonderful week, and I feel like I should say something awesome before you go. Go out and make the magic happen. All right. See you guys. Bye.